The second research line I work is medical imaging for biomedical systems, which includes functional brain mapping for epilepsy and pre-surgical mapping of brain tumors, machine learning and dynamic causal modeling of neuroimaging data. And the third research line I work is artificial intelligence for neuroimaging, which includes diagnostic models for disease classification and a correspondence between human and artificial vision models. In terms of research methods, I study human behavior with paradigms from experimental cognitive psychology. I use electroencephalography and magnetoencephalography to study the temporal dynamics and combine it with multivariate decoding of cognitive processes. For spatial mapping, I use functional MRI and I combine it with multivariate decoding and computational modeling. And I also use deep neural network modeling for correspondence between biological and artificial system. In my today's talk, it will be mainly focused on human vision. So I myself hate boring talks, and it seems like I'm just talking to a computer now. So uh, now I will show you an image, and you guys have to recognize some objects in that, right? And then I will ask, what did you see? So what I just showed you is an image. Okay, can anyone tell me what did you see?
It's a market.
Okay,
Yeah.
anyone else? You guys need to speak up. I mean, I hate just talking to a computer, right? So, okay, I think now it's too much time. I just gave you. Okay. So what you just saw is, as Alvin already mentioned, it's a supermarket. That's a layout of the scene. There are different objects which are present in the scene, right? And there are some text, and there is a spatial layout as well. You already know that the objects in a supermarket, for example vegetables, they will be lying on the lower side of visual field. However, if there is a big banner of a shop, that will be on the top side. This means human perception is incredibly efficient for effortlessly performing complex visual tasks like object recognition, category recognition, scene recognition, and visual search, and that too in very complex natural environments. This efficiency is explained by canonical computations in the visual system, and I will today focus on two such computations. First is prediction, which allows the visual system to predict future states of the world from its past states, and sorry, and integration, which allows the visual system to meaningfully group information to facilitate the perception of complex scenes. Now, first, I will talk about predictions. And before we dive into predictions, I will first explain predictive processing. Predictive processing is a fundamental cognitive mechanism accounting for how the brain interacts with the external environment through perceptual inferences. And this predictive processing theory in neuroscience is mainly taken from linear predictive coding in digital signal processing. The basic idea was when you transfer data from one frame, one position, one location to another location, rather than sending each and every frame, what you can do, you can take the difference, like what the difference between previous and the current frame along with the location, and you can send that data. That was really well accepted and like quite popular technique in digital signal processing. Then this idea was taken up, and the first model of predictive coding of retina that came in 1982, where they showed that the light intensity of the next frame could be predicted from the previous frame. And then again, this model was implemented to the human cortex by Rajesh Rao and Billard in 1999. That was one of the most influential paper at that time, which claimed that the brain has two streams of information processing. One is a top-down processing, another is a bottom-up sensory processing. However, the problem with this model was that it claimed that all the cells in the human brain, they both encode prediction and prediction errors, which is biologically not possible. And similarly, there were some other models, for example predictive coding biased computation, which says that there is a division process which is performed by the cells, but till date we don't know like how cells perform such kind of division. A very interesting explanation came in 2005 by Dr. Carl Friston from UCL, who claimed that there are two types of cells in the human brain, one which encode for predictions and another which encode for prediction errors. And that's the model which is quite popular now and is widely accepted. Just to give you an idea, whenever there is a sensory stimulation, be it be from human eyes, ears, tactile, olfactory, any stimulus, the brain is a generative model which is not reactive, it's proactive. That means it always keep on training itself and predicting what would be the next output. So it makes a sensory prediction, and now you have an actual sensory input through sensory stimulation, and you have the prediction made by the human brain. So the difference between both of them is termed as prediction error. This prediction error is again used for the adaptation, and the internal generative model is updated to make more precise prediction for the next incoming stimuli. So there are two main streams. One is called top down descending activations, and again is bottom up ascending activations, right? So this is mainly pre-stimulus before you see any stimulus, and this is process is done like post stimulus. So the our interaction with environment is dynamic in nature. Whatever we see around us have mainly two types of information. It has information around the immediate surrounding, which is called what information, that tells us what are the features of our surroundings, right? Here in this example, the feature could be like a painting, the color of the wall, the desk, all those things, right? So the content or in I mean while all the objects are grouped together, we can call it also call it like a context. And it has another type of information which is called when information, which is tells about the temporal structure of the events. When I'm talking to you, my speech has a temporal dimension. When you listen to music, it has a temporal dimension. The world around us is not pictures, right? It's just some sort of video which is going on all the time, so it has a temporal dimension. And since humans are capable of performing such complex tasks, as I show in this video, this requires a precise estimation of both what information and when information. Here are like two examples that what happens when you wrongly estimate the temporal information in our day-to-day life, right? So there have been several paradigms to study predictive processing. The first one and the very popular ones are oddball designs. So oddball design is basically you present the same stimuli again and again time, and suddenly there is something which is a odd stimulus. For example, here there were like a toaster, 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 and then again a rooster comes up. So this is oddball experiment here. However, the other case could be equiprobable sequence where every stimulus is presented the same times. Right? This is quite a popular oddball paradigm. And another paradigms were basically rowing paradigms where you present a series of standard stimuli, then a deviant stimuli, and this again this deviant becomes the new standard now. So it keeps on going. Then there is standard. Again, you present a deviant stimuli, and then the deviant becomes the standard. So here you understand how does the brain go through adaptation when there is something which is not expected. How does the brain treat that, and how does it adjust, making it the new standard stimuli. There were again motion based paradigms where you the motion of predicted dots could be either predictable, which is like a linear motion, or it could be a random sequence, which is not predictable. And then it came like the expectation based paradigms. Expectation based paradigms is when you present something to your participants, you generate a queue, and this queue means that there is 75% probability of the occurrence of a tiger compared to a 25% probability of a horse, right? So when you see this queue, that means you will expect a tiger, but if you are presented with a horse, this will give you an like error type signal. However, if there could be a second queue where there is 50% probability for both the stimulus. So far, like these expectation based paradigms are one of the best paradigms which could explain the human brain functioning. And in predictive coding framework, expectation suppression imply that activity in stimulus selective neurons is reduced when the perceptual expectations of an observer is fulfilled. For example, in this case, like the blue curve, which is for the expected, has a lower amplitude compared to the unexpected case here. right? So till now, the open questions in the domain were: how is expectation suppression from the context, that is the what information, is affected by the timing? And how is expectation suppression affecting the decoding of stimulus selective features? To answer the questions, we had like open in the domain. I used magnetoencephalography. How many of you know about magnetoencephalography? Anyone? Am I audible?
Yes,
Yes,
you
sir.
are.
Okay. So here, I mean, most of you must be aware of electroencephalography, which measures the electrical activity in the human brain. But all the electrical impulses in the brain, they do have a magnetic field. And magnetoencephalography is such a sophisticated machine which could measure the magnetic field changes happening in the human brain. And this is really a very sophisticated machine and quite rare as well. So just to give you a context, in whole India we have just two magnetoencephalography machines. And to answer the questions, we use these artificial stimuli which are called Gabor patches. So these Gabor patches has couple of features, and we manipulated two features of Gabor patches. First, the orientation angle, that is how tilt the angle is. This could be a 0 degree angle, this could be a 90 degree, this could be a 60 degree angle, again a 90 degree, right? And another is cycles per degree, also called as spatial frequency, that how thin or thick these lines are. If you were one of my MEG participants, you could have experienced something like this in the scan. What you just saw was a sequence which began always with a cross fixation, and there were total of 5 Gabor patches. The first 4 being called as entrainers, and the last one is one out of these 4, which is called as the target. So we have mainly done 3 manipulations. First, the orientation angle of the target could be either predictable or unpredictable from the entrainer sequence. For example, in this given case, the entrainer 1 has an orientation angle of 30 degree, then it becomes 0, minus 30, minus 60, and if I ask you to predict the target, that would be minus 90 degree angle, right? The second manipulation we did, sorry, the second manipulation we did is the timing between these Gabor patches. In case of predictable, they have always a constant timing of 200 milliseconds between all of them and a long gap of 600 milliseconds before the target. In case of predictable, it could be randomly varying between 70 to 330 milliseconds, right? And the third manipulation we did was basically we tried to delineate the overlapping and confound of attention and prediction. So if you, both these processes, attention and the predictions, are top-down processes. So to delineate it, we created our orthogonal design where the predictive feature, like the prediction, is on one feature, that was orientation angle. However, the participants, they were asked to report the spatial frequency of the target, which could either be higher or lower compared to the internal sequence. All 4 of them had same spatial frequency. Now, if we define all the, if we do different manipulations, we had 4 different conditions. The first condition is called what plus when condition, where both the orientation angle of the target as well as the timing between these Gabors is fixed or predictable, right? The second condition is the what condition, where the orientation angle is predictable, but the timing between them is not predictable. The third condition is when, where the orientation angle is not predictable, but the timing between the Gabors is predictable. And the fourth condition, which was a random condition, where neither the orientation angle nor the timing between the Gabors is predictable, right? This is the experimental paradigm, and rest of the thing I will be talking about will be based on this. So if anyone has any doubt, This is the time to ask. Please feel free to ask, and I will be happy to explain it again. Okay, since nobody is asking, I understand everyone got everything. So yeah, after this, we recorded MEG from a couple of participants, and we plot here. What you see is the activity from occipital channels, which are close to visual areas. And first, we need to verify that there is some sort of expectation suppression going on. So what you see in the blue curve experiences more suppression, which is what plus one condition, that is the where orientation angle and both timing is predictable, compared to the case where orientation is not predictable. Again, when we compare what where orientation is predictable compared to the random where it's not predictable, we again see a suppressing response as we move across entrainers and up to the target. But the question was, how does the timing that affects, how does when affects the context, that is what? To do so, we compared the condition what plus when with the what condition, where the orientation angle is predictable, but in case the timing is not predictable, another case the timing is predictable. And the results show that temporal prediction enhances the expectation suppression. So I would also like to bring your attention to this very initial response. If you look here in the beginning, when we presented first Gabor, there were no differences in this very initial baseline. At the target, again, the difference vanishes. But within the sequence, there is a big difference in this very initial response. This means that in case of temporal uncertainty, the brain relies more on its internal generative model compared to the predictable temporal phases. And when we combine this MEG data with MRI and try to source localize this differential effect, we found the effect arising from Brodmann area 18, which is V2, a higher order area compared to a V2. Now, from here, you can see that there is some sort of suppression going on. But what is the brain suppression? Like is it some sort of relevant information? Is it suppressing the noise? What is the brain suppressing here? Now, to understand, before diving into the results, I would quickly take you through a technique called temporal decoding. Suppose if I want to understand how does the brain processes houses and faces, at what time point in their processing hierarchy do the processing starts to diverge, right? At what time point the difference between the representation of houses and faces is completely different. So to do so, what I can do, I will put any of you as a participant, and either in EEG or MEG, and would throw a lot of faces and houses on your stimulus screen. So the data I will have will be of three dimension, right? First, it will have number of trials, that is how many times I presented faces and houses. Number of channels, depending on if you use MEG, you would have a 306 channels. If you have EEG, you can go from 16 channels to up to 128 channels, depending on your EEG configuration. And the time for how long you recorded it. So uh, if I take, I mean, strip this three-dimensional structure into two-dimensional structure for each and every time point. So for one time point, I will have all the channels and all the trials. If I throw this data to any linear decoder, right, I will have a decoding accuracy at one time point. Again, I take the second data set and I have another decoding accuracy. I can loop it over time to decode it at all the time points, and you will see like there is a decoding accuracy which is presented as a curve here. So here you see that uh, before even we presented, the classifier is jittering around 50%. There are slightly up and down activity, which is just the classifier is just decoding the noise. And as soon as close to 100 milliseconds, that is the classical N100 response, the processing between human faces and houses started to begin. And close to 170 milliseconds, that is the classical N170 response, right? We see the peak decoding accuracy. Now, compared to the classical univariate evoked responses, where we don't know like what's happening after the peak effect, right? You can say that here that there is a dip in the processing hierarchy, and again there is some recurrent processing going on, which is basically the fine tuning of the representations. And the representations are sustained up to like 1000 milliseconds. That means what you just saw, the brain will keep its memory up to like brain will be processing it up to the next 1000 milliseconds, right? So we use the same technique to the data, to our data, and we mainly trained two classifiers. The first classifier, to train between the orientation angle of the target. So whether it's horizontal versus vertical, we trained it on each and every entrainer and the target. And we trained another decoder to decode the spatial frequency, which is cycles per degree, which was a high CPD versus low CPD. Now here, what we found here, the first finding was in case of temporal uncertainty, because here it's what plus when with what only, the representations are short-lived compared to temporal predictable scenario. We can also claim that the information about the target orientation was available even before the target was presented. That means even before 400 milliseconds, right, the brain knew in case of predictable that what will be the next target here. And you can see that the decoding accuracy was significant here. These dot lines are like statistical significance of these effects compared to the chance level. And it starts dropping when you have the task associated with the target. This is another finding which claims that the attention, once you divert your attention, that attenuates the prediction the brain was making at that particular time. And like just to show you here, you see that the orientation angle which started to become predictable from antenna 3, right, the decoding accuracy about the target, it started to increase from E3 E4, and it's again significantly decodable at the target. We have we can also see that the feature which was task related, right here, this was represented much stronger. Like the decoding accuracy, the peak decoding accuracy is close to 97-98%, close to the task irrelevant feature, which is represented around 78 to 79%, which means that the features which are task relevant, they are represented more strongly compared to the task irrelevant features. And just to show the robustness of our method, spatial frequency or CPD, which was same across all the four trainers, we don't have any differential effect here. I mean, there is some noise decoding, but nothing is significant here. So I will quickly summarize the results of this part of my presentation. It shows that expectation suppression of evoked responses was more prominent, that is larger, when the timing of a trainer was unpredictable. Even though neural signals were suppressed, decodable information was enhanced. That means the brain is suppressing the noise and somehow fine tuning the representations about the incoming stimuli. Now, this was all about prediction part. Now I will talk about integration and how it's related to the visual preference. In day-to-day life, we go to different places, come across a lot of scenes. Some of these scenes appear to be more beautiful than the another one. Of course, there are cognitive factors that relate to the evaluation of pictures, such as emotional associations, but research has shown that the visual properties such as color, contrast, symmetry of scenes are also capable of explaining these differences. A very interesting a very interesting finding came from visual processing theory. But till date, we lack an understanding of overarching principles in perceptual processing that shape aesthetic preference. And processing fluency theory, it says that the ease with which a stimulus can be analyzed in the visual system plays a critical role for the perceived beauty. In past, there have been several candidates used to quantify fluency, such as symmetry and integration. Today, we will only talk about integration. What is spatial integration? On left side of the screen, you see that there is a scene, right? There is a car present here, a road, and a person. And if most of you must recognize the scene, and you would say that this person is asking lift from the car driver, right? If I split this into different participants, different objects of this scene, this is a person, right? He is just having his thumb, maybe for anything, we don't know. A car, we don't know where it's driving, and the road, which appears to be empty, right? So if we take the representations for the whole and again measure the representations for individual elements through any neuroimaging modality, let's say fMRI, right? And we do a correlation of whole versus sum of its part, right? We will obtain a correlation value. If this correlation value is lower, that means there is high amount of integration present in the image. And conversely, if there is this correlation value is higher, right, that means there is low amount of integration present in the image. I would like to iterate this again, right? So if correlation between the whole and its sum of its individual elements is lower, that means there is high amount of integration present in the image. And conversely, if the correlation is higher, that means the sum the whole is very similar to its sum of parts, there is an amount of very less integration happening in the image. This has been demonstrated through like monkey studies and human studies, and is a classical principle in computational neuroscience. Now, the open questions again in the domain were: can integrative processing in human visual cortex predict perceived beauty or visual preference? If yes, what types of feature are integrated? Does the brain integrate the low level features, mid level features, or high level features? And very interesting one, does artificial vision models, that is deep neural networks, integrate similar to the biological model, which is the human brain? So first, I will show you results from a deep neural network. We used a deep neural network as a computational model of vision, and there were mainly two reasons to use a deep neural network. First, so far, they have been considered to be the one of the best models mimicking the human brain functioning. And second, DNNs also give us an infinite amount of testing capacity. We can test as many as images as we want. A DNN won't complain like a human participant after 2 hours that I am tired now and I don't want to continue the experiment, right? This gives us a lot of computational power. So we used a VGG16 network, which was trained on Places 365 data, which is a collection of 1.3 or maybe 1.6 billion images scenes. And there could be two approaches to use a deep neural network. First, you just fed the network with an image, and the network will do some sort of like computations and will give you an output that this is an ocean, right? That's one approach to use deep neural networks. Another very interesting approach could be to see what's happening within the network, irrespective of the decoding accuracy and all those things, right? We see how what's happening in the layers of DNN. So what we do here, we fed the network with an whole image, and to uh, introduce integration for deep neural networks, we divided the whole into two complementary halves using a checkerboard pattern. So one half is complementary to each other. We fed this image to the network, this image to the network, take the representations in each and every layer, and if we correlate the representations for the whole with some of its two parts, again we will have the correlation. And similar to human experiments, if this correlation value is lower, there is high amount of integration present in the image, and the other way around, if this correlation is high, the integration is lower. And to quantify for degree of integration, we used 5 different spatial scales, which could be a 2 by 2 scale, 4 by 4, 8 by 8, 16 by 16, and 32 by 32. This scale shows that how many parts are presented at one time. So one half of this would be just two quadrants, another half would be these two quadrants, right? Now, first, we did a couple of behavioral studies, right, to link the human behavior to the human brain and the deep neural network data. So the first study we did is we presented a total of 250 natural scenes to 25 participants, and we asked them to rate it how beautiful or not beautiful this image is. And we presented this for a very brief exposure, only 50 milliseconds. Previous work has shown that all observers can judge the beauty of an image even under such brief presentation regimes. And we wanted to test the same in our experiment as well. So, what you just see here is a plot which shows the correlation between the integration measure derived from the deep neural network, right, as I shown on the last slide, and the beauty ratings, which is sum of all 25 participants we have, and we do a correlation of integration measure with the beauty, right? On x-axis, you have all the activity from all the 16 layers of VGG16 network, and all these different lines correspond to which spatial scale it belong to. These dots here shows a statistical significance. What so what we just see here is correlating the integration measure with beauty rating revealed a strong relationship between integration and perceived beauty, with correlations as high as zero point 0.6. Those who work with the brain behavior correlations, like they must be knowing this kind of correlation value is really, really high in terms of brain behavior correlations. And correlations were strongest in intermediate to late layers of the deep neural network, suggesting that integration over mid and high level features determines perceived beauty. Correlations were apparent across all spatial scales, but were strongest for coarser scale with a decrease before 16 by 16 and 32 by 32 scale. Now, this is one kind of analysis, right? Another approach could be we can test how well the network is predicting the beauty ratings, right? So what we did is we fitted a linear model with all the 16 layers of network included as predictor for a beauty rating for all the images except one image. Let's call it like a left out image. So we train the model with the response for 249 images and then test it on left out images. And if we do this for each and every image, so we will have like 250 ratings, right, which are predicted by the network, and the real beauty ratings which are predicted by the human, right? And if we do a correlation, we see again a very strong correspondence, very strong correlation between human ratings and predicted ratings by the network. This suggests that integration over larger parts of image is a stronger predictor of perceived beauty than integration across fine details. So this was the first study we did, right? Then we did another 25 participant who rated the same 250 natural scenes which we used in the first experiment, but this time we gave them unlimited amount of time for response, allowing them to also cognitively evaluate the image in detail. But surprisingly, the integration measure essentially predicted perceived beauty equally well as briefly presented images. This means that beauty of unnatural scenes is already computed in the brain at very, very early stages of perceptual processing. When we did this linear model fitting, we have a very similar trend. Now, this time we did a third experiment now, and this time we changed the image set, right? We tested completely new set of 250 natural scenes and rated for their beauty by again 26 observers. But this time we also asked them to rate the order, like how orderly this image is arranged and how simple or complex this natural scene is, along with how beautiful or not beautiful the natural scene is. Here also, we found integrative processing is a very strong predictor for perceived beauty. And the human ratings of complexity and order do not fully capture the visual features that predisposes integrative processing in deep neural networks. Again, while fitting the linear model, a very similar trend here. Right. Now, then we wanted to test if integrative processing measure could predict perceived beauty not only for natural scenes but also for a wide range of photographs that depicts objects, people, and everyday situation. So we used OSIS database, which is available online, and it has been rated by a lot of human observers for their beauty. Now, given the large variability in content and emotional valence, the question was, can our integrative processing measure still predict perceived beauty? We expected that predictions derived from our integration measure would be reduced because there is very high amount of variability in the content and emotional valence. As we hypothesized, correlations value were indeed lower, but the deep neural network, if you see the statistical significance here, right, DNN derived measure still predicted perceived beauty for novel images. Again, for this linear model, we found that the trend is similar with the lower correlation values. Now, to explore how the relationship of integration and perceived beauty changes when visual properties are stripped away in targeted ways. We manipulated the original input image supplied to a DNN in target images. I mean, I was expecting somebody should have asked this question that the kind of effects you are having, this could be this could be driven by one particular image feature, right? So what we did is Do you remember this 8 by 8 scale, spatial scale, which has the highest correlation, right, in the first study? So we use this as a reference, and first we decolorize the image. We wanted to see if the color of an image is it driving the integration. The answer is no, because after decolorizing, we did the whole process again, right? Did the whole experiment again with the deep neural network, and it shows a very similar integration value. Again, we did a luminance matching. So we matched the luminance across all the stimuli and see if the integration measure could still predict the perceived beauty. Again, we did a histogram matching. So they matched the whole, all the 25 images, right? And then as a test, we change the pixel values. So you distort the scene using changing the value of randomly changing the value of all the pixels in a natural scene. So an original image looks something like this here, and this in this case it couldn't predict. That means when there is something completely, there is no context here, right, then the integration measure is not working, which is showing the robustness of the analysis. Then we did a high pass filter, low pass filter, right, a 90-degree flip. We just flipped all the images by 90 degree. We flipped all the images by 180-degree, right, and did a jumbling. So even with all these manipulations, we found that mid-level and high-level features could still predict perceived beauty even after all these changes. That means integration is, sorry, not driven by any of these particular features. Not only for one study, we tested this for study 2, which we did, uh, study 3, and study 4, which was the overseas database. So this visual charting shows that both very basic visual features like color, luminance, and more high-level configuration properties thus cannot account for the interrelationship of integration and perceived beauty, suggesting that features of intermediate complexity are critical for the prediction of perceived beauty. Now, based upon the results from 4 behavioral studies, which I presented, it's evident that the DNN derived integration measure successfully predicted the beauty ratings for natural images. However, we cannot be fully sure whether our DNN indeed replicates the way in which human brain integrates information across images. After all, DNNs have been shown to share features from human visual processing in potentially critical ways. So, time to do some human experiments, right? So we did a fMRI study with humans, and as I mentioned earlier, right, with humans it's not possible to test as many as images as we want, as we are limited with time, right? So we only tested one spatial scale, which was 2 by 2, with human participants. So we show this whole image to the participants, first half, second half. We take the neural representations for the whole and correlate the neural representations for its two complementary sum of its two complementary halves, and again do the correlation. As I have already explained this several times, right? If the correlation value is lower, there is high amount of integration present in the image. Conversely, if there is high correlation, that means there is low amount of integration present in the image. So we take the activity in 7 different brain regions, which is V1, V2, V3, and V4, which are called as early visual areas, and the scene selective cortex, which is OPA, NPA, and PPA. Here, integrative processing in scene selective cortex, that is PPA, predicts the aesthetic appeal of natural images. Now, to show the correspondence between correspondence between the activity in the PPA and the activity in DN. So we correlated both of them, and what we find here that there is a very high correspondence in mid-level features between human, integrative processing in humans and the integrative processing in deep neural networks, showing a very revealing a correspondence between integration in biological and artificial vision. So again, just to iterate, like fMRI results show integrative processing in scene selective cortex, specifically in PPA, predicts the aesthetic appeal of natural images. Right now, this work I have shown till now is already published. You can go and if you look for my profile, you will find this paper. Now I'm presenting some of the work which is still going on. So what we did now, we only work with natural scenes and photographs. To do a follow-up study to test integrative processing in artworks. There are artworks which has different kind of features and people perceive them beautiful. So we did two studies, right? In the first study, we had two types of artworks. The abstract ones, which are just random images irrespective of like giving or showing any context. I'm sorry. And the second one were representational artworks, which depicted some sort of scenes, right? And again, to quantify for degree of integration, we have these all five different spatial scales. Here, for the abstract artworks, the DNN, which was trained on natural scenes, when we do the correlation between integration from deep neural networks and beauty ratings from humans, it couldn't predict, right? However, for the representational artworks, it predicts very well. That means integration among humans only like predicts the perceived beauty rather than visual features that are not meaningful. Now, this was the first study we did, right? We did another study, which is where we use the Vienna like art picture system database, which consisted of 999 artworks divided into five broader categories. The first category is landscape, which is very similar to a natural scene, right? Second is still lifes, which are more similar to like something which is like a living object. Then the third category was social scenes. Social scenes where a group of people depict action which is happening in the scene. Then we had portraits, which are simply like human faces. And the fifth category is towards abstraction. So this one is not completely abstract artworks, right? They are towards abstraction. So this is different from what I showed in the study one. First, we took all these 999 artworks together to compute the correlation between integration measure and perceived beauty. On the group level, it shows that again mid and late layers of the network has very high correlation values. This is for all these five category were already combined, right? We did another analysis by just computing integration for individual category. So what we found is, yeah, first we did for landscape, and this shows very significant correlations across mid and late layers, right? For social scenes, I mean, for still life, it again shows, I mean, it again shows like significant correlations. But for social scenes, right, the DNN could not predict the perceived beauty. Again, similarly for the portraits. So, one of the explanations we have at the moment for this one is that these networks are not trained on human faces, they are mainly trained on like natural scenes. Now we are testing it with the networks, deep neural networks, which are trained on faces, and then we would be able to make some conclusion about integration in human faces, right? And this is like very interesting one. So for towards abstraction, if you see like the early and mid layers, right, they doesn't predict anything. However, the late layers, right, they show significant correlation. That means integration only in late layers of DNN could predict the perceived beauty. That means these artworks are not completely abstract. By the end, there is some sort of association, right? And the DNNs are picking up those associations. So just to like give you a summary, integration in artificial and biological vision determines perceived beauty ratings across different stimuli. That is, we use different sets of stimulus and different task demands. We tested the effect of timing, right? A brief presentation time versus unlimited presentation time. And integration marks as a computational principle that predisposes the aesthetic appeal of natural images and artworks. So, on this note, I would like to thank my supervisors. First, Professor Daniel Kaiser, who was my postdoc supervisor, and all the work on integration was done with him only. I want to acknowledge Professor Nicola Molinero, who was my main PhD supervisor, and Professor Matthew Borgignon, who was my PhD co-supervisor, and Professor Radosla Kichi, who was again my third PhD supervisor. I am grateful to Mineco, which is like the Ministry of like Economics for Spain for funding my PhD, and thankful to EMBO short-term fellowship for providing me an opportunity to visit Radosla Kichi's lab, and the Adaptive Mind for funding my postdoc. I would also like to thank all my lab mates who have made my life easy while living abroad. So here are a couple of interesting moments we all had together. And the final one is, as you all know, I have recently joined IIT Mandi. This is the scenic view of IIT Mandi, right? So in rainy season, you see something like this. I'm currently looking for MTech research scholars and PhD scholars. So we have a program which is called MTech Research. This is not the classical MTech program. Here, the students they only do coursework for 6 months, and rest of the time they spend their thesis working on a project, and again PhD scholar. So the key domains I'm looking on is someone having expertise in biomedical signal and image processing, cognitive and computational neuroscience, and AI/ML for healthcare. So if anyone is interested in these areas, please contact Dr. Alvin, and he can forward your CV, and I will have a look. Yeah, on this note, I would like to thank you all, and I'm really happy to answer any questions. In case there is a question you don't want to ask here, please send me an email on these email IDs, and I will try to respond as soon as possible. Thank you so much. Thanks a lot.
Thank you, Dr. Sanjeev, for the wonderful presentation. Any question from audience? You can unmute and ask questions to Dr. Sanjeev. If nobody have any question, I'll ask one question. How did you choose those images, Dr. Sanjeev? All these images which you have shown.
Uh-huh.
How did you choose for your research? The different buildings, abstracts.
Right. So these data, these images are taken from Places 365 dataset, which is a huge data set for natural scenes. It has a close to like 1. I don't remember the exact figures, but should be close to 1.5 billion images of natural scenes, and which are extended up to 1,000 categories. So one category could be different images of buildings. Another category could be parking lots, mountains, rivers. So that's a huge data set, and we have picked different categories from these Places 365 data set.
Okay, okay. It's kind of a random, or you want to choose this particular image and ask for the
No, there were different categories, but we mainly choose based on the living categories versus non-living categories. For example, a building would be non-living, however, trees, all those things could be like considered as living ones, and there could be natural versus man-made. So there are different categories you can, again, subcategories you can divide. So we take a very broad categories.
Thank you, sir, Dr. Sanjeev. Any more questions from audience? If no, we will wind up this session. Thank you. Thanks once again, Dr. Sanjeev, for the wonderful session.
Thank you so much for inviting me. It was really nice to have a talk.
Thank you. Thank you. Thank you, sir.
Alvin, should I leave?
Yes, yes, please.
Okay, thank you.
Thank you, sir. Thank you.